Welcome, everybody. Thank you so much for your time this morning. I am delighted to be presenting on this topic today. Before I get started, I just wanted to introduce myself. My name is Anastasia, and I am a clinician educator at Genova, Europe. And like many of you, I'm also a registered nutritional therapist and recently a certified functional medicine practitioner. And so testing is such an important part of my practice to help guide me in the decision-making of my therapeutic protocols. So today's focus will be on Genova's essential metabolic fatty acid panel, which can be done in either as an individual test or as part of the NutriVal, which I'm sure many of you will already know about. And this is something that I will go briefly into when talking about testing options. So my aim is to be presenting for around 30 to 40 minutes, after which there will be time for a Q&A at the end. And if you haven't already used the GoToWebinar software, you can ask your questions written in the GoToWebinar control panel under the questions tab. You will also see a support guide under the handouts tab as well, which you'll notice is actually the NutriVal support guide. However, the information on this test and the biomarkers included in this test can be found in one of the support guide's key chapters. So here is the outline of today's presentation. So initially I'm going to go over the outline of Genova's test and what we're actually measuring and how. And then I'll go through each of the biomarkers in the test so you can get a comprehensive overview of what the test measures and why. And then I will move on to the inflammatory cascade and fatty acid metabolism. As well as that, the clinical applications. So just starting off with an overview of Genova's panel and how we test the fatty acids. So a key intro into the test is how we actually measure fatty acids. And as I mentioned, the test is available as a standalone, which is measured as a blood spot, or as part of the NutriVal, which is a full blood draw. Now, the blood draw measures fatty acids as a weighted percentage of the cell membrane, and the blood spot, which is as a standalone, or I should add also as an add-on to the Metabolomics Plus, which is another one of our tests, includes the red blood cell and plasma. So what's the difference between the two? So as I mentioned, the fatty acids in the blood draw are measured in the red blood cells as a weighted percentage of the cell membrane. And this means that when dealing with percentages, each fatty acid can of course influence others. So for example, if a client or a patient of yours is supplementing with omega-3 or fish oil, for example, that will of course increase the overall omega-3 percentage, which may lower the omega-6 percentage. As I'm sure many of you will already know, the red blood cell life averages 90 to 120 days. So red blood cells will of course naturally reflect a longer status than plasma. Now, blood spot evaluation is whole blood, and that encompasses both red blood cell and plasma. So this may reflect both short and longer term status. Though I should note that in internal data that we've carried out, there is very good correlation between the two. So just before I move on to the other stuff and the biomarkers and why we test those biomarkers, I just wanted to go over a little bit of a biology recap into what actually is a fatty acid. So it's helpful to be reminded of what a fatty acid actually is and how it is named based on its structure. So a fatty acid is a carbon backbone with a carboxyl group at one end and a methyl group at the other. And it's the number of the carbons which delineates the fatty acid as short chain, medium chain, long chain, or very long chain. And then in terms of saturation, the presence or absence of double bonds between the carbons reflects the degree of saturation. So when the carbon backbone contains no double bonds, it's called saturated. And unsaturated fatty acids contain one or more double bonds within that carbon backbone. So here is the main page of the panel. So we can see here all of the analytes within their reference ranges. And you'll also note at the bottom right-hand corner of this page, you have some ratio biomarkers, which can give us insights into various things like cardiovascular risk, as well as the functioning of certain enzymes like the delta-6 desaturase enzyme, which I will of course go into later and why we do that. We also display the omega-3 and omega-6 analytes in this convenient visual layout of the pathways related to fatty acid metabolism and the respective enzymes that are used for conversions to occur within the metabolism of omega-3s and omega-6s. So the elongase and the desaturase enzymes, which as you'll see are listed here in the middle of the fatty acid metabolism chart, they essentially convert essential fatty acids into others by adding carbon molecules to the backbone or by inserting double bonds. And as you can see, each enzyme has its respective cofactor nutrients needed to help propel the functioning of the enzyme. So it's useful to see here where someone may be struggling in terms of conversion pathways. And as you can see, this is a sample report. So the level of the fatty acid is outlined by the color-coded system here. So green, of course, indicates a normal level, red, significantly high or low, depending on what direction the triangle is pointing in, and then yellow, moderately high or low. So the other thing that is worth mentioning is that omega-3 and omega-6 fatty acids are competitive in their use of these enzymes. And so we may often see a fatty acid that is dominating an enzyme. This is why it's really useful to have these results outlined on this visual page here. So I'm just going to go into a breakdown of the metabolic function of each of the biomarkers that we test, and then I'll move on to the more clinical sort of applications. So omega-3 really needs no introduction. Most of you will of course know that omega-3 fatty acids are very highly anti-inflammatory and are used in cell membrane production, function, and overall gene expression. Most Western diets are deficient, of course, in common omega-3 food sources like oily fish, flax, chia seeds, and deficiencies in omega-3 have been correlated with many clinical conditions, some of which are things like neurodevelopmental and behavioral disorders, cardiovascular disease, cognitive decline, mood disorders, and many others. Now, as you can see, the reference ranges for omega-3 fatty acids are one-tailed. So what this means is that there isn't an upper level of the reference range for each of these analytes. And that's because their health benefits are so well studied and deficiencies are associated with so many clinical conditions. So generally speaking, the higher the better. And then you also see that there is a total percentage of omega-3s as well at the very bottom. And that's added by, that's calculated by adding each of those analytes together. And you'll see that for each of the fatty acids that we test, you also get a percentage at the end. So then we have the omega-6 fatty acids, and a breakdown of the omega-6 analytes are closely evaluated in relation to omega-3. Of course, you've seen that on the second page with the fatty acid metabolism layout. Now, many of you will know that, of course, omega-6 is very much related to its pro-inflammatory properties. And of course, throughout evolution, the dietary intake of omega-3 and omega-6 polyunsaturated fatty acids has proportionately changed from a ratio of 1:1 to 20:1 or more in favor of omega-6 fatty acids. And this shift has coincided with an epidemic of obesity, metabolic dysfunction, and many other significant clinical implications. As I've mentioned, one of the main concerns about omega-6 fatty acids revolves around one of the downstream metabolites, arachidonic acid, which is a precursor for the inflammatory cascade, and it's this potential pro-inflammatory nature of omega-6s, as well as their susceptibility to oxidation, which can lead to many clinically deleterious effects. Lastly, they also compete, as I mentioned before, with the omega-3 cascade for the use of the elongase and the delta desaturase enzymes. So you may find them dominating the enzymes and therefore leading to lower levels of the omega-3 metabolites like EPA and DHA. So, of course, we mainly associate omega-6 fatty acids with their pro-inflammatory nature, but it should also be emphasized that not all omega-6 fatty acids are concerning, and in fact, some have important physiological impacts and can be also highly anti-inflammatory depending on the analyte. We then have the omega-9 fatty acids, which we measure mainly as oleic acid, which is found highly in foods like olive oil, as well as certain nuts. And this fatty acid is particularly important in cell membrane fluidity. It also has been shown to have anti-inflammatory and immunomodulatory effects, which have been extensively studied and found to be beneficial in many conditions like cancer, neurodegenerative disorders, inflammatory disorders, autoimmunity, and the list goes on. I should also mention that oleic acid has also been shown to lower blood lipids, mainly total cholesterol, LDL cholesterol, and triglycerides, and has been shown to dampen the inflammatory response within the vascular endothelium. And then we have the saturated fatty acids, which compared to unsaturated fatty acids, as I'm sure many of you will know, have a higher heat index and a better oxidative quality, making them ideal as cooking oil at high temperatures. Now, their structure having no hydrogen bonds contributes to a lack of cell membrane fluidity. And it's worth noting that some saturated fatty acids can be synthesized in the body from carbohydrates. And so attempts to lower saturated fatty acid levels by removing dietary sources should, of course, encompass the strategy of limiting carbohydrates. Lastly, short-chain fatty acids, saturated fatty acids have been shown to alter lipid metabolism and elevate cholesterol levels, which I'm sure many of you will know. And they are also involved in the inflammatory cascade, which I'll be going through towards the end. However, we also know that saturated fatty acids may play an important role in hormone production, gene transcription, cellular membrane structure, and protein signaling. So, of course, they do play their important roles too. We then have the monounsaturated fatty acids as omega-7 fatty acids. Now, there are many good properties to these as well. They can decrease the oxidative susceptibility of LDL. They also can decrease platelet aggregation and increase fibrinolysis, which protects against things like thrombogenesis. And then lastly, we have the trans fats, which are particularly measured as oleic acid, which I'll go through at the end. And these have been shown to induce oxidative stress and alter mitochondrial signaling. And they are highly pro-inflammatory. So we'll be going into the specifics around the trans fats and why they are pro-inflammatory in terms of what their specific impacts are on the inflammatory cascade. But of course, we typically imagine that the exposure to trans fats nowadays aren't such a risk factor, considering that there are now very strict labeling rules, particularly in the UK, for hydrogenated fats and shortening in processed foods. However, in those that I have seen with elevated levels of trans fats, the intakes of foods like crackers, and these are often crackers that contain very refined carbohydrates and refined vegetable oils, also biscuits and other baked goods, In those that eat those foods in abundance, I have seen high levels of trans fats. So there is a clear correlation there, and it can often be seen as well, even in today's, very strict labeling rules, etc. So as mentioned initially, we also display some important ratios to demonstrate various aspects of health, one of which is the delta-6 desaturase enzyme activity. And here we're looking at the ratio between linoleic acid and DGLA, two key omega-6 fatty acids. Now, the two key enzymatic steps that are required to synthesize DGLA from the essential linoleic acid are the delta-6 desaturase enzymes and the elongase enzymes. But we're predominantly measuring the functioning of the delta-6 desaturase, as it has been shown to be a more influential enzyme in the conversion to DGLA. Now, the reason why we assess this enzyme is down to the importance of DGLA as a potent anti-inflammatory omega-6 fatty acid, which is dependent on the intake of GLA, or gamma-linolenic acid, and of course the functioning of the delta-6 desaturase enzyme. Now, I will of course go into the properties of DGLA and how to increase it, but it's worth noting that the functioning of this enzyme is often proposed as a functional biomarker of zinc status. So despite the fact that there are several vitamin and mineral cofactors that are required for each enzyme, which are listed there in the fatty acid metabolism chart, the zinc deficiency can have a huge impact on the activity of delta-6 desaturase. And of course, in addition to that, we also know that zinc deficiency can impact things like fatty acid absorption, oxidation, and incorporation into red blood cells. So having an impaired delta-6 desaturase activity level can point to zinc insufficiency as an important cause. And then other considerations to include, of course, the lack of the other vitamin and mineral cofactors, insulin dysregulation as well. In order to increase DGLA, which I'll go into towards the end as well, many clinicians do use things like evening primrose oil, borage oil, and blackcurrant seed oil to bypass the delta-6 desaturase enzyme, though of course cofactors for elongase should also be optimized as well. And then you will see that the inability to convert the linoleic acid to DGLA is often associated with things like diabetes, atopic dermatitis, rheumatoid arthritis, cancer, and cardiovascular disease. So it is a very important ratio to analyze in the context of many inflammatory conditions. So then we have the cardiovascular risk ratios, where we are assessing the balance between omega-3 to omega-6 metabolites. So we have the omega-3 to omega-6 ratio, which broadly speaking looks at all of the metabolites of each fatty acid in relation to each other. And then we also have the arachidonic acid to EPA ratio. And of course, we know that both of these metabolites, they compete for the use of the delta-5 desaturase enzyme to be synthesized. And there are many diseases that are related to having high levels of arachidonic acid, such as cardiovascular disease, mood disorders, and cancer. We then have the omega-3 index, which is defined as the red blood cell percentage sum of EPA and DHA, both of which are important anti-inflammatory omega-3 fatty acids. Now, this particular index was first proposed in 2004 as a cardiovascular risk factor. And since then, it has been repeatedly verified as an important cardiovascular biomarker and studied in the context of other diseases like obesity, insulin resistance, and mood disorders. A reasonable target for the omega-3 index is 8% and upwards to decrease disease risk. And generally speaking, from experience looking at many of these tests, what I often see that is struggling is not so much the EPA, but actually the DHA. And in those that I have seen, that have healthy omega-3 indexes, they are often supplementing omega-3 and eating omega-3 rich sources such as oily fish. So it's actually quite a hard level to achieve just by a regular diet without eating those foods and without supplementing. Okay, so I'm going to move on to the inflammatory cascade specifically and how fatty acid metabolism can influence that. So how do essential fatty acids influence inflammation? So it's all about the eicosanoids, and I'm sure many of you would have studied this extensively. So what are the eicosanoids? I've listed a few of them here: prostaglandins, lipoxins, thromboxanes, leukotrienes. Essentially what they are is they constitute a large and expanding family of lipid signaling molecules, which are key mediators and regulators of inflammation and immunity, and are generated from omega-3 and omega-6 fatty acids through enzymes like the COX, LOX, and the cytochrome P450 enzymes, and I'll go through that in more detail in the other slides. So what are they triggered by? They are essentially activated by different stimuli like trauma, ischemia, pathogens, and pro-inflammatory cytokines. And they have many different roles. Eicosanoids are essentially converted from membrane phospholipids to provide an efficient way for cells to respond to various stimuli that require a cellular response. And these are things like causing inflammation, pain, and fever, reducing blood clotting and cleaning up unnecessary blood clots, dilating blood vessels to increase blood flow, the contraction and relaxation of specific muscles, including the gut and airways, depending on where the prostaglandins are being produced. They also control ovulation, the menstrual cycle, and they help to induce labor by inducing the contraction of smooth muscles in the uterus. So they are very, their roles are very diverse. And it's really helpful to understand how eicosanoids are actually produced to grasp why they're so important in the inflammatory cascade. So here we have a diagram of the arachidonic acid cascade, which is courtesy of the Institute of Functional Medicine. And this is where the pro-inflammatory eicosanoids are generated. As you can see, at the very top, we have the cell membrane, which is where we find the fatty acids like omega-6s and omega-3s. And when there is a trigger of some kind or a stimuli like those that we saw on the previous page, let's say a pathogen, the enzyme phospholipase A2 is activated, and that draws out the arachidonic acid from the cellular membrane, releases it from the cellular membrane, and then that goes into the various enzymes for conversion into the eicosanoids that are needed depending on the stimuli. So as you can see, you've got the COX-1, the COX-2, and the 5-LOX, which are the various enzymes, and then you've got the respective eicosanoids. These eicosanoids then go on to amplify those inflammatory signals to recruit things like leukocytes, cytokines, and immune cells to aid in things like pathogen resistance and clearance. So now many of you will be most likely familiar with some of the COX and LOX enzymes and some of the main inhibitory medications that are commonly used to prevent the pro-inflammatory sort of prostaglandin production of those COX and LOX enzymes. So things like ibuprofen and aspirin will act. Their main function is to inhibit the function of COX and LOX enzymes, and that of course inhibits the production of pro-inflammatory prostaglandin synthesis, and of course that then results in decreased pain, inflammation, and fever. So you can start to see really how modulating omega-3 and omega-6 ratios within the diet, within supplementation, within the red blood cell can have a profound impact on influencing the inflammatory cascade, and of course reducing levels of arachidonic acid within the cell membrane can help to prevent the overstimulation of pro-inflammatory prostaglandins. So we have here, oops, let's go back a slide. So we also have the omega-3 generated eicosanoids. We predominantly know that eicosanoids derived from omega-6 fatty acids have pro-inflammatory and immunoactive functions, whereas eicosanoids derived from omega-3 fatty acids, like EPA and DHA, for example, have anti-inflammatory properties, which are traditionally attributed to their ability to inhibit the formation of omega-6 derived eicosanoids. So as you can see here, EPA, DHA, and DGLA, so DGLA, remember, is not actually an omega-3 fatty acid, it's the anti-inflammatory omega-6 fatty acid. All three are able to block the COX-1, COX-2, and LOX pro-inflammatory mediators. We also know that the eicosanoids produced by fatty acids like EPA and DHA are lipoxins and resolvins, which act to control the proliferation of immune cells, as well as other things like cancer cells, by inhibiting growth-promoting pathways. They control the synthesis and release of pro-inflammatory cytokines and can prevent inflammatory cytokines like interleukin-8, as well as other potent inflammation signaling molecules like NF-kappa B. And you'll also note that DGLA, as I mentioned before, is a potent COX and LOX inhibitor. So this is predominantly why it's such an important omega-6 fatty acid to have healthy levels of, and why we assess the delta-6 desaturase enzyme. So here are some of the key functions of the resolvins and the lipoxins. So as I mentioned, they inhibit growth-promoting pathways, particularly related to cancer. They can suppress the release of pro-inflammatory cytokines, particularly blocking TNF-alpha signaling, which is a very potent pro-inflammatory cytokine. They can also increase NRF2 levels, which is a potent transcription factor that upregulates genes that combat oxidative stress and help to increase levels of antioxidant proteins and reduce levels of reactive oxygen species. They also help in the migration of neutrophils to sites of inflammation. And of course, neutrophils also need to be cleared from the site of inflammation in a timely way after acting on a particular pathogen. The balance in the neutrophil life cycle is essential to avoid damage to the tissues. And the delayed apoptosis of neutrophils is one of the major causes of diseases like cancer, for example. And lipoxins, what they do is they help in the clearing of the neutrophils from the site of infection, and they help to resolve the inflammation. So they are very important, in terms of their function in preventing the over-perpetuation of inflammation. So I'm going to finally move on to the clinical applications. So as you've seen on the previous slides, this is the second page of the report, which is a useful way to assess fatty acid imbalances from a bird's eye point of view. As I've mentioned a few times, omega-3 and omega-6 fatty acids compete for the same enzymes. And this is really how the diet and supplementation can effectively prevent the overproduction of pro-inflammatory eicosanoids. So the consumption of EPA through oily fish as well as supplementation can effectively decrease the amount of arachidonic acid present. And that's because they compete for the same enzyme, the delta-5 desaturase enzyme. The intake of oils like gamma-linolenic acid-rich oils, such as eating primrose oil, blackcurrant seed oil, borage oil, also result in increased levels of DGLA through the elongase conversion. And I should say that both EPA and DGLA compete with AA, or arachidonic acid, for the production of eicosanoids. So that's really one of the key functions, key reasons why or how our diet and supplementation can really reduce the inflammatory load. So gamma-linolenic acid is synthesized from linoleic acid through the delta-6 desaturase enzyme, and this enzymatic reaction is very slow and is further impaired in vitamin and mineral deficiency, such as zinc, which is something I've already highlighted. There are other things that can impair the functioning of that enzyme, And that's things like stress, smoking, the intake of alcohol, and systemic inflammatory conditions, which can further slow down the conversion. So since the synthesis of GLA is not efficient, the dietary intake of GLA is super important, and that can be done through the intake of organ meats, as well as the supplementation of products like borage oil and so forth. The supplementation of things like borage oil, for example, have been studied in effective treatment for many conditions such as rheumatoid arthritis, dermatitis, and things like also diabetic neuropathy. So they've been shown to decrease inflammation and have other important health impacts like improving bone health. They help to regulate lipid metabolism and also have been shown to have beneficial impacts on the skin. But really, the clinical importance of GLA is in its rapid conversion to its downstream fatty acid, DGLA, which is, as we know now, anti-inflammatory. Now, there is a bit of a concern of GLA supplementation leading to rapid conversion through DGLA down into arachidonic acid, but there have been some clinically researched ways to help mitigate this. So here is an expanded slide on this particular concern and what we can do to prevent this. So I've put here a screenshot of a study in terms of research that shows that by supplementing EPA at the same time as GLA, this can help to mitigate the effects of rapid conversion to AA, since there is this enzymatic competition for the delta-5 desaturase enzyme, which is responsible for both arachidonic acid production and EPA metabolism. Now, in this particular study that I got this screenshot from, and you'll see it referenced in the next slide, they used 3 grams of GLA and in combination with 3 grams of EPA, so fairly high doses. And I've given here a list of common supplements that are GLA rich, and as you can see, borage oil has the largest percentage compared to other sources. So here are some of the potent anti-inflammatory impacts of combining GLA and EPA together. So supplementing GLA has been shown to markedly decrease the capacity of inflammatory cells like neutrophils to produce leukotrienes. And the likely inhibitory mechanism of GLA really stems by its capacity to be elongated by the endogenous elongase activity within the neutrophil to DGLA, which is the anti-inflammatory omega-6 analyte. And that, of course, is anti-inflammatory in nature because it blocks the functioning of the COX and LOX enzymes and therefore the pro-inflammatory prostaglandin synthesis. And the addition of EPA supplementation prevents the accumulation of arachidonic acid in response to GLA supplementation. And it does this without inhibiting the accumulation of DGLA in neutrophils. So this is why it's so such a potent combination. So how much omega-3 do we supplement in clinical use? So studies in anti-aging, in the anti-aging impacts of omega-3 supplementation have demonstrated efficacy of a total daily intake of 1.5 grams of EPA and 1 gram of DHA. That's a combined total of 2.5 grams of omega-3 as a concentrated dose. And in one particular study, they saw that after 4 weeks of administration of this dose, there were significantly lower levels of interleukin-6, interleukin-1 beta, and TNF-alpha. And one of the interesting things is that even greater effects were seen with longer therapy over a longer period of time. Now, tissue levels of omega-3 fatty acids reflect dietary intake, of course, but quantitative data about the rate of the incorporation and levels as a function of intake within the red blood cell are actually quite scarce. But there are some studies that show that EPA levels in erythrocyte membranes don't actually plateau until the supplementation of between 6 to 12 months, which suggests that the greatest lowering effect on inflammatory mediators may not actually be seen until therapy has been administered for several months. So it just goes to show that just supplementing for a few months at a time actually that there may still be some benefits there, but potentially we're going to see even better, even more sustainable impacts over a longer period of time with more consistency. So we then move on to the trans fats, and I wanted to expand a little bit more on the impact of trans fats on the inflammatory cascade. So as I mentioned in the beginning, trans fats induce oxidative stress and they can alter mitochondrial signaling. And once incorporated into plasma membranes, they actually activate NF-kappa B and induce adhesion molecules, which contributes to endothelial dysfunction and, of course, inflammation. They also reduce the availability of nitric oxide, which further contributes to endothelial dysfunction. And they also promote fat storage in the liver, which contributes to conditions like non-alcoholic fatty liver disease. It's unsurprising that they have, of course, been implicated in many conditions like cancer, cardiovascular disease, insulin resistance, and so forth. And the intake of industrially produced trans fats, and that's a key word, industrially produced, because we also find trans fats naturally occurring in dairy and meat products. The intake of industrially produced trans fats are positively associated with systemic concentrations of various inflammatory markers like C-reactive protein, tumor necrosis factor, and interleukin-6. I should note that within Genova's panel, we assess Linolenic acid, which is the principal and most abundant trans fatty acid in the Western diet. And as I mentioned before, it's found in partially hydrogenated vegetable oils, and there's also trace amounts of naturally occurring Linolenic acid in meat and dairy products, ruminant animals, which of course are naturally occurring. So what about saturated fatty acids, which is of course another key part of the panel? So the key distinction to make is that different saturated fats exhibit different functions and properties. And we measure palmitic and stearic acid, which are the main two dietary saturated fats. And then we also measure the very long and odd chain fatty acids, which exhibit completely different properties. So for example, very long chain fatty acids and odd chain fatty acids demonstrate distinct beneficial properties compared to palmitic and stearic acid. And there have been several meta-analyses that suggest a beneficial association of very long chain saturated fatty acids with cardiovascular health outcomes, as well as lower risks of type 2 diabetes, atrial fibrillation, heart failure, and coronary disease. And these very long saturated fatty acids may also be important in neurodevelopment and cognition. Now, very long chain fatty acids are actually mainly found in food sources like nuts, seeds, and pulses. So it's not found often in dairy and animal protein. So in terms of palmitic acid and its effect on the inflammatory cascade, when we're looking at palmitic acid, there are some distinct influencing factors in relation to the inflammatory cascade that are important to highlight. So as the name suggests, it is a major component of palm oil, but it can also be found in meat, dairy, cocoa butter, coconut oil, and olive oil. Palm oil and palmitic acid are also found in many products, from skincare products, cereals, baked goods, etc. Now, the key thing to understand when we're looking at palmitic acid and its potential to become inflammatory is in situations of uncontrolled endogenous synthesis. So dietary intake of palmitic acid is counterbalanced by the endogenous production of palmitic acid according to or depending on the physiological needs of the specific tissue or the nutritional factors. Regardless of palmitic acid dietary intake, the body makes it as needed. And research shows that it is in situations of uncontrolled endogenous synthesis of palmitic acid that it can start to have pro-inflammatory properties. So for example, it has been shown to enhance NF-kappa B. It has been shown to induce the mRNA expression of COX-2 enzyme, and also increase levels of interleukin-1 beta, interleukin-6, and TNF-alpha. And it is an excessive intake of carbohydrates and a sedentary lifestyle where we see a disruption in the homeostatic balance of palmitic acid, which can result in dyslipidemia, hyperglycemia, and also increased fat accumulation, particularly ectopic fat accumulation, which relates to the deposition of triglycerides within cells of non-adipose tissue that normally contain only small amounts of fat. So this all leads to lipotoxicity, as well as altered immune responses, and of course a stimulation of the inflammatory cascade. And the disruption of this balance is implicated in many diseases. So we have here a list of a few of them: type 2 diabetes, cardiovascular disease, and also cancer risk. And then we have stearic acid. So stearic acid is mainly abundant in animal fat, but it also can be seen highly in things like cocoa butter and shea butter. So it's often used in things like detergents, soaps, cosmetics, shampoos, shaving cream, for example, and it can also be synthesized in the body from palmitic acid. So when assessing levels of stearic acid in this test, it can also be important to assess the patient's use of cosmetic products which might have stearic acid, and of course their absorption of that. So stearic acid alone doesn't seem to exert such negative impacts compared to other saturated fatty acids. So for example, it's not a strong substrate to make triglycerides, and it hasn't been shown to raise LDL cholesterol. But the intracellular accumulation of both palmitic acid and stearic acid have been reported to induce the activation of NF-kappa B and the secretion of pro-inflammatory mediators, and there are some associations with cardiovascular disease. And then finally, we have the odd chain fatty acids, which, as you can see, they have an odd number of the carbon molecules, 23, 15, 17. Now, for years, it has been concluded that odd chain fatty acids or saturated fatty acids were of little significance in human health. However, there is now a realization that they are in fact relevant and important physiologically speaking. So they mainly originate from dairy. The human body can also synthesize them by elongating propionic acid, which is a short chain fatty acid formed in the microbiome with the fermentation of fiber. And they have been shown to increase cell membrane fluidity, actually more than polyunsaturated fatty acids, and are actually being studied as a result of this as a form of treatment for Alzheimer's disease. I should also say that there is positive associations in epidemiological studies between levels of odd chain saturated fatty acids and reduced risk of inflammation, cardiometabolic disease, multiple sclerosis, and also non-alcoholic steatohepatitis, so non-alcoholic fatty liver disease, basically. They are also being studied as adjuvant therapies in cancer due to their cell signaling properties, which induce targeted apoptosis. And as I mentioned, increased levels are seen in diets that are high in fiber. So intake of fiber can actually increase odd chain fatty acids levels at the expense of other saturated fatty acids. And some studies suggest that odd chain fatty acid levels may actually act as a biomarker for dietary fiber intake. And then finally, increased levels are also seen in diets that are high in dairy as well. Okay, brilliant. So that's the end of the presentation. I'm going to move on to, oops, what am I doing? I'm going to move on